Today I will discuss about gene regulation. Gene regulation. Now I will divide this topic gene regulation into three parts. The first part will be the definition of gene regulation. The second part will be why we will read this gene regulation, and the third will be the mechanism of gene regulation. So the first question is, what is gene regulation? Second one is why we will read gene regulation, or why is it important to read gene regulation? Why is it important to read gene regulation? The third question is how gene regulation occurs in the cell, means the mechanism of gene regulation in a cell. The mechanism of gene regulation in a cell. So at first we will discuss what is gene regulation. Now what is gene regulation? Gene regulation is the method to control the expression of gene at a different time, at a different place, or how much amount a gene should be expressed, that all it regulates in a cell. So if we write the definition of gene regulation, then it should be it is the process that is used to control at first timing, second location, third quantity or amount, of expression of a gene in a cell. So this is the definition of gene regulation. Next we will discuss about why is it important to read this gene regulation process. Second is why is it important to read about gene regulation in a cell. The first and foremost important to read about gene regulation in a cell is, suppose here is cell A, here is cell B. Now the each and every cell of our body has a same genetic makeup, but what it differs from cell A to cell B or a cell A to cell C. It only differs from the expression of gene at a spatio-temporal manner. Means, suppose some gene X is expressed in A at a particular time, but this is silenced in B or C. Means that is different from B and C. So how it takes place? Now we will discuss the mechanism how it takes place. Mechanism of gene regulation. If we see the mechanism of gene regulation, there are lots of gene regulation process. At first, we have to talk about post-transcriptional gene regulation process. Post-transcriptional gene regulation. Second one is post-translation. This occurs most of the time in a cell. This is a rare event, so we will only discuss about post-transcriptional gene regulation in my following topic. So, post-transcriptional gene regulation. There are different types of post-transcriptional gene regulation. We will only discuss about the RNA mechanism through which there are control of gene expression in a post-transcriptional manner. So we will only discuss about RNA interference mechanism. Now, what is RNA interference? It is sometimes abbreviated as RNAi. Now, RNA interference means RNA is interfering to something. Now we will discuss what is interfered by this RNA, and this RNA will interfere to whom. Now, if we will discuss about the definition of RNA interference, then we will see that definition: it is a evolutionary conjunct conserved mechanism of gene silencing which is induced by small silencing RNAs. Now, evolutionary conserved means this mechanism is seen from east to all higher mammals. So if we will divide these small silencing RNAs in a different topics, so we will only now focus to these small silencing RNAs. Small silencing RNAs. This is of two types: siRNAs, mRNAs. Now we will focus only this siRNAs or siRNA mediated RNAi mechanism. So before talking about siRNA mediated RNA interference mechanism, we have to discuss about some features of siRNAs. The first and foremost important feature is these are non-coding RNAs. The second important feature is it induces gene silencing in a sequence specific manner. So from this second feature, we can say that this siRNAs will degrade only those messenger RNA which are perfectly complementary for these siRNAs. Now we will discuss how siRNAs help to silence a gene. The first step is processing of precursor long double-stranded RNA. Now we will schematically show you that how this processing will happen. Suppose this is a double-stranded RNA which has 5 prime, 3 prime, 3 prime, 5 prime. This is long double-stranded RNA. Now here dicer will bind to this double-stranded RNA, and it will process this long double-stranded RNA to form various or lots of short stretches of RNA. Those are called siRNAs. And this siRNA should possess 2,3' overhanging region, and this overhanging region have only 2 nucleotides. Let's see this diagram. After the processed by dicer, these siRNAs will form like this is 5 prime, this is 3 prime, this is 3 prime, this is 5 prime. So there are 2,3 prime overhang region. Here is one, and here is another one, and these 2,3 prime overhang region possess only 2 nucleotides, and this is called siRNA. Now after the formation through the processing of long double-stranded RNA, now this siRNA will be bounded by RNA binding protein. Let's see this through the next diagram. Before going to the next diagram, we will have to discuss some features about dicer. What is dicer? Dicer is an endonuclease that have 3 domains. The first domain is ATPH. Look at this. This is dicer. Look at the smile face. It has 3 hands. It has 3 hands. And one hand each. This is ATPH, RNA helicase, RNA helicase domain. This smile face is dicer. Although he is a devil one that will help to degrade a messenger RNA. Now this one is catalytic RNase 3 domain. This one is double-stranded RNA binding domain. So he or she is alien which has three hands. In one hand it will bind with the help of ATPase RNA helicase domain that will help to provide energy during the processing of the long double-stranded RNA. This domain enhances the processing reaction, and this domain will help to bind the dicer with the double-stranded RNA. Now we will discuss about the formation of risk loading complex. Second is formation of risk loading complex. What is risk? Let's discuss about it later. So, after the formation of siRNA, that is 5 prime, 3 prime, 3 prime, 5 prime. This is siRNA. Here, the siRNA is already bounded. RNA binding protein is driven bound. This complex is called risk loading complex. So, this complex is called risk loading complex. This is RNA binding protein. RNA binding protein. This is dicer. This is dicer. So after the formation of this loading complex, now we will discuss about how this loading complex will help to reach this complex to the risk. Now I think you all are very curious about to know that what is risk. So let's discuss about risk. Simply, it means RNA induced silencing complex. This is the graveyard of messenger RNAs, where messenger RNAs are lost. After the formation of risk loading complex, then here comes the role of siRNAs. This siRNAs will, if we will see that these siRNAs have two strands, 5 prime, 3 prime, 3 prime, 5 prime. This is called the guide strand, which will guide to the target messenger RNA, and this is anti-guide or simply passenger strand. Although in further method this passenger strand will be loosed or will be lost. Now this is the now here is risk, here is risk loading complex. Risk loading complex. This is RNA binding protein. This is the dicer, and now here here argonaute protein will bind. Here argonaute protein AGO2. This argonaute protein is also an endonuclease that will cleave the passenger strand, and then we will left only the guide strand. So if we draw a schematic picture where there is only guide strand with the RIS complex, this is RIS complex, this is my guide strand, 3 prime, 5 prime, 5 prime, 2, 3 prime, this is my guide strand. Now this guide strand will bind to the target messenger RNA. Here is target messenger RNA. And the argonaute protein, which is the most important component of this RIS complex, will degrade the messenger RNA. And after the degradation of messenger RNA, there will be a recycling process. Suppose after the degradation of messenger RNA, there is left only guide RNA and the RIS complex. This is the guide RNA. This is the RIS complex. Now it will further complementary to the different, or suppose there is a lots of messenger RNA, so it will bind to another messenger RNA and will degrade them and so on. This is the whole machinery or whole mechanism through which siRNA mediated RNA interference occurs. This is seen in every eukaryotes, like from yeast to mammals. So this is a most important post-transcriptional gene regulation mechanism through which a cell is revived through a virus attack. Now at last I have to discuss about the most important thing is that this siRNAs should have nearly equal to 21 nucleotides, and these siRNAs are perfectly matched or perfectly complementary to the messenger RNA or their target messenger RNA. So that is all about how siRNAs mediated RNA interference occurs in a cell.